All right, let's dive right into this explainer. We're going to explore how modern genomic data literally dismantled classical taxonomy and completely redrew the eukaryotic tree of life. If you're studying biology at the undergraduate level, you've definitely realized by now that the simple categories we were taught as kids just don't hold up under the DNA sequencer. So today we're looking at the hard molecular data that forced scientists to basically rewrite the textbooks.
So our story actually starts over 300 years ago with the Dutch scientist Antony van Leeuwenhoek. He famously wrote, "No more pleasant sight has met my eye than this, of so many thousands of living creatures in one small drop of water." He was looking at what we kind of just lumped together as protists. And for centuries, biologists did exactly what he did. They just looked. They categorized these microscopic marvels purely based on their visual morphology, whether they creeped along with blob-like appendages or looked like tiny trumpets.
But this shift from categorizing organisms by how they look to uncovering their true relationships hidden deep within their DNA, that was a massive game changer. Visual morphology relies on physical traits like flagella or cellular structures. Metagenomics, on the other hand, classifies organisms through whole genome sequencing and molecular data. We've totally moved from an era of biological observation into an era of hardcore molecular data analysis.
So here is our roadmap for today. We'll start with 1, the fall of Kingdom Protista, 2, the rRNA endosymbiosis evidence, 3, complex plastid evolution, 4, supergroups defined by DNA, and finally 5, rooting the eukaryotic tree. Let's get right to it.
Section 1, the fall of Kingdom Protista. Let's look at how the whole illusion of this kingdom completely fell apart under the scrutiny of modern metagenomics.
You see, for a long time, Kingdom Protista was essentially just a taxonomic junk drawer. If a eukaryote wasn't a plant, an animal, or a fungus, it just got thrown in there. But then, metagenomic studies revealed this incredible treasure trove of unknown diversity. We found eukaryotes that are incredibly tiny, literally just 0.5 to 2 micrometers in diameter. That is exactly the size of many prokaryotes. It proved once and for all that visual size and shape are absolutely terrible tools for classification. Some of these tiny organisms actually turned out to be more closely related to you and me than they are to other protists. So the whole Kingdom Protista just had to be abandoned.
Moving on to Section 2, the rRNA endosymbiosis evidence. Since we obviously can't trust our eyes, we have to turn to molecular data, which brings us to a huge breakthrough regarding the origins of the mitochondria.
To trace the origin of mitochondria, researchers needed a highly conserved genetic sequence, right? Well, because ribosomal RNA, or rRNA, performs such critical fundamental cell functions, it's under intense selective pressure. It mutates incredibly slowly over millions of years, making it basically the perfect molecular clock. Researchers cloned and aligned 617 nucleotide positions from the small subunit rRNA to act like a genetic time machine, comparing a eukaryotic mitochondrion against major clades of bacteria.
Let's actually analyze the data matrix from that research. These numbers represent the percentage of the 617 nucleotide positions where the pair of organisms have the exact same base. Notice that massive 48% match between the wheat mitochondrion and the alpha proteobacterium agrobacterium tumefaciens. Compare that to the much lower 38 and 35% matches with the other bacteria. The data is practically screaming at us here.
So what does this specific genetic overlap actually prove about the very first complex cells? I mean, why on earth does a wheat mitochondrion share so much sequence with a specific alpha proteobacterium? Well, it's the smoking gun for endosymbiosis. It proves that an early eukaryotic host cell literally engulfed an ancient alpha proteobacterium, and instead of digesting it, they formed this incredible symbiotic relationship. And that engulfed bacterium eventually evolved into the mitochondrion powering all eukaryotes today.
All right, Section 3, complex plastid evolution. Now we're going to build on that idea and look at how these early cells acquired the ability to photosynthesize, because the story definitely doesn't end with mitochondria.
This definition is key to wrapping our heads around the staggering complexity of cellular evolution. Primary endosymbiosis gave us the first plastids when a eukaryote engulfed a cyanobacterium. But secondary endosymbiosis, that is like molecular inception. It's a heterotrophic eukaryote engulfing a photosynthetic eukaryote that already has an endosymbiont inside it.
By tracking these exact steps, molecular biologists found undeniable proof hiding right inside modern cells. Take chloroarachniophytes, for instance. Inside their cells, researchers actually found a tiny vestigial nucleus, called a nucleomorph, just sitting right inside their plastid. And the DNA sequences transcribed from this nucleomorph indicate that the engulfed cell was a green alga. It is literally a fossilized genome nested inside another cell, giving us a perfect molecular roadmap of how entirely new lineages were formed. Which brings us to Section 4, supergroups defined by DNA. So, with the old kingdom Protista completely dismantled, and endosymbiosis making cellular lineage incredibly complex, scientists had to redraw the entire map of life.
And that leaves us with 4 major eukaryotic supergroups: Excavata, SAR, Archaeoplastida, and Uniconda. It's absolutely crucial to understand that a supergroup like SAR, which is Stramenophiles, alveolates, and rhizarians, is defined almost entirely by whole genome DNA similarities. If you looked at them under a microscope, there is no way you'd guess they're related. Their unity is written exclusively in their genetic code.
To put into perspective why this matters on a global scale, just look at the diatoms. They're a really critical photosynthetic group within the SAR supergroup, encased in these unique glass-like walls of silicon dioxide. During massive population blooms, their numbers get so unbelievably vast that when they die, they sink directly to the ocean floor before decomposers can even break them down. This literally pumps massive amounts of carbon dioxide straight out of the atmosphere to the ocean floor. It's a perfect example of how microscopic genetically distinct clades exert enormous control over our planet's carbon cycle. Finally, Section 5, rooting the eukaryotic tree. Now we arrive at the climax of our molecular detective story, trying to figure out which of these supergroups actually diverged first. To solve this giant puzzle, researchers utilized a rare event approach. They hunted for a highly unlikely evolutionary mutation involving 2 specific genes, one coding for the enzyme DHFR and the other for TS. In bacteria, animals, and fungi, which represents the ancestral condition, these are entirely separate genes. But researchers discovered this incredible derived character, a super rare evolutionary event where the DHFR and TS genes physically fused into a single gene coding for a single protein. Because gene fusion is such an incredibly rare event, it's highly unlikely it happened independently multiple times. And get this, Excavata, SAR, and Archaeoplastida all share this fused gene, but Uniconta doesn't. This implies that the first 3 form a clade sharing a more recent common ancestor with each other than any of them does with Uniconta. So, the data analysis leads us to a thrilling, albeit heavily debated conclusion. The Unicons may have been the very first eukaryotic supergroup to branch off from the rest of complex life.
To wrap things up, let's zoom in on a really fascinating unicont, Dictyostelium discoideum, the cellular slime mold. It's used as a model organism to study multicellularity, and it totally blurs the line of what an individual even is. Usually, they're solitary single-celled amoebas, but when food gets scarce, hundreds of them chemically signal each other and aggregate into a multicellular slug. Eventually, they form a fruiting body where some cells intentionally die to form a stalk, literally sacrificing themselves so the cells at the top can survive and reproduce as spores. But here's the catch. Molecular studies show that a mutation in a single gene can turn a cell into a cheater. A cell that joins the aggregate but refuses to die for the stalk. If cheating's an evolutionary advantage, why don't they all just cheat? It turns out non-cheating cells actually have a surface protein that recognizes other non-cheaters, completely freezing out the mutants. It leaves us with this final provocative thought. The molecular rules of complex life are incredibly fragile. The very existence of multicellular life relies on these invisible, ancient recognition systems to prevent cheating at the cellular level. So what other invisible biological contracts are written right into our DNA?